Let's start by understanding antigen-antibody reactions. These are fundamental interactions in immunology, where specific antibodies bind to specific antigens. This reaction is the basis for many diagnostic tests, especially in serology, where blood samples are examined to detect immune responses. The presence of bacteria and cellular materials reminds us that these reactions happen in complex biological environments, and the blood samples collected are crucial for analysis. Now, let's clarify what an antigen-antibody reaction really means in serology. It's when a specific antigen combines with its matching antibody under the right conditions, like optimal pH, temperature, and electrolytes, and this combination becomes observable. This specificity is what allows us to detect precise immune responses reliably in laboratory tests. What makes antigen-antibody reactions so unique? Well, they can happen naturally inside the body or be recreated in the lab. The reaction is highly specific, involving the whole molecules rather than fragments. Importantly, neither the antigen nor antibody gets denatured during the process, and the binding can be reversible depending on how strong the molecule's affinity and avidity are. This reversibility is key to understanding how immune complexes behave. So, what happens after the antigen and antibody react? Several outcomes are possible. They might form visible precipitants or agglutinates, kill or lyse cells like T cells, neutralize toxins or fix complement proteins. Sometimes they can immobilize motile bacteria. Notably, the reaction itself doesn't denature the molecules, which means the immune complexes remained intact and functional. Let's explore antigenic determinants, which are the specific parts of an antigen molecule that antibodies recognize and bind to. Some antigens have just one determinant, while others have multiple, arranged in pairs, rings, or complex branching structures. These variations affect how antibodies interact with antigens, influencing the strength and specificity of the immune response. In other words, the more determinants, the more complex the potential interactions. The prozone phenomenon is a fascinating aspect of antigen-antibody reactions. It occurs when there's an excess of antibody relative to antigen, preventing proper lattice formation and thus reducing precipitation. As antigen is added, antibody precipitation increases until it reaches an equivalence point, then decreases again if antigen becomes excessive. Understanding this helps prevent false negative results in serological tests by recognizing the zones of antibody excess, equivalence, and antigen excess. There are several types of serological tests that utilize antigen-antibody reactions. These include precipitation and agglutination tests, immunoelectrophoresis, Western blot, complement fixation, immunofluorescence, and various immunoassays. Each test type offers different advantages depending on the target antigen or antibody and the clinical context, allowing us to detect and measure immune responses with precision. Precipitation reactions involve soluble antigens and antibodies forming insoluble complexes that appear as floccules. These reactions can occur in different media, like liquids or gels, and they rely on lattice formation where antigens bind to antibody FAB sites. This visible precipitate indicates a positive reaction, making precipitation tests useful in identifying specific immune complexes in samples. To give you some examples of precipitation reactions, slide precipitation, like the VDRL test, tube precipitation, such as the Kahn test, and diffusion methods, including single and double diffusion in 1 or 2 dimensions. These techniques differ in how antigen and antibody diffuse and interact, but all rely on the formation of visible precipitant bands or lines to confirm the presence of specific immune reactions. Let's talk about single and double diffusion techniques. In single diffusion, antigen diffuses through a medium containing antibody, forming a precipitant band where they meet. Double diffusion adds an extra layer, allowing both antigen and antibody to diffuse towards each other, creating a more defined precipitin line. These methods help us analyze antigen-antibody interactions qualitatively and quantitatively in a controlled environment. Building on that, single and double diffusion can also be performed in 2 dimensions. In single diffusion in 2 dimensions, antigen diffuses radially into an antibody containing agar, forming a ring of precipitation. Double diffusion in 2 dimensions involves multiple wells containing antigens and antibodies, creating precipitin lines where they intersect. These setups allow for detailed analysis of antigen-antibody specificity and cross-reactivity. Immunoelectrophoresis combines electrophoresis and immunodiffusion to separate and identify proteins based on their electric charge. Molecules migrate towards electrodes depending on their charge, forming bands that represent different antigens. This method includes variations like countercurrent electrophoresis and rocket electrophoresis, each providing unique ways to analyze immune complexes and protein concentrations. Countercurrent electrophoresis is a technique where antigens and antibodies migrate towards each other under an electric field within a gel. Antigens move towards the negative electrode, while antibodies move towards the positive electrode. When they meet, they form precipitin bands, making it a useful method to detect specific antigen-antibody interactions quickly and with clear visual results. Immunoelectrophoresis separates antigens by charge and then introduces antibodies to form precipitation arcs, revealing antigen identity and concentration. Rocket electrophoresis, on the other hand, produces sharp, rocket-shaped bands whose height correlates with antigen concentration. These methods provide detailed profiles of immune proteins, which are valuable in diagnostics and research. Agglutination reactions involve insoluble antigens and antibodies forming visible clumps or aggregates. This reaction is often more sensitive than precipitation tests because the clumping can be seen with the naked eye. Agglutination is widely used in blood typing and infectious disease diagnosis due to its clarity and reliability. Examples of agglutination tests include slide agglutination, like the Widal test, tube agglutination, such as CRP and RF tests, hemagglutination used in blood typing, passive agglutination like RPR, co-agglutination for salmonella screening, and the Coombs test. Each serves specific purposes in detecting antibodies or antigens in clinical samples. The direct Coombs test detects antibodies attached directly to red blood cells, useful for diagnosing immune-mediated hemolytic anemia. The indirect Coombs test detects antibodies in serum that can bind to donor red blood cells, important in blood transfusion compatibility. Both tests rely on agglutination caused by anti-human antibodies linking red blood cells, indicating the presence of antibodies against red cell antigens. Immunofluorescence tests use fluorescent dyes attached to antibodies to detect specific antigens in cells. Direct immunofluorescence involves a labeled antibody binding directly to the antigen, while indirect immunofluorescence uses a secondary labeled antibody to detect a primary antibody bound to the antigen. These tests are especially useful in diagnosing autoimmune disorders, offering sensitive and specific detection. ELISA assays come in three main types: indirect, sandwich, and competitive. Indirect ELISA detects antibodies binding to antigen-coded wells, sandwich ELISA captures antigen between two antibodies, and competitive ELISA measures antigen by competition with labeled antigen. All involve enzyme-linked antibodies producing a color change that quantifies the antigen or antibody present, making ELISA a versatile and powerful diagnostic tool. The complement fixation test detects antibodies by their ability to fix complement in the presence of antigen. If antibodies are present, complement binds to the antigen-antibody complex, preventing lysis of sheep red blood cells, indicating a positive result. If no antibodies are present, complement remains free and lyses the red blood cells, showing a negative result. This test helps confirm the presence of specific antibodies in patient serum. Finally, let's clarify some key terminologies. Sensitivity is a test's ability to detect even small amounts of a substance, minimizing false negatives, which is crucial for preliminary screening. Specificity refers to detecting the substance accurately without cross-reactions, minimizing false positives, important for confirming diagnosis. Understanding these helps us evaluate and choose the right serological tests for accurate results.